with power.
Welcome to the Deep Dive. Today, we're really getting into the weeds of drug metabolism.
That's right.
And not just what it is, but how it's fundamentally changed how we discover drugs. It's gone from just fixing problems to really driving the whole process.
Absolutely.
Now, this deep dive is especially for you if you're preparing for your PhD qualifying exams in pharmaceutical sciences. We know biosynthesis and drug metabolism are huge topics. So our mission today is to take a pretty dense chapter and break it down, make it clear, give you those lecture-ready insights, not just what happens, but why it matters, with real examples you could use yourself.
Exactly. And we're drawing from Scott Grossman's Drug Metabolism and Drug Design and Development. It's a foundational text.
Okay.
And what's really fascinating is seeing how the field has, just exploded, but the core goals, they're still the same.
Which are?
Basically figuring out how a drug behaves in the body, understanding its disposition, that whole journey, absorption, distribution, metabolism, excretion, and, how it gets chemically changed along the way.
Okay, so let's talk about that explosion. I mean, back in the day, you had giants like R.T. Williams taking maybe 6 months, right? Just to map out a drug's disposition.
Yeah, unbelievable now.
It feels like a different world compared to today's tech and knowledge base. So what's the impact? What does it mean for the biotransformation scientist working now?
Well, their role is just much broader, more integrated, I'd say. They're not just observing how a drug behaves, they're linking that behavior directly to safety and efficacy.
Right.
And here's the interesting part, historically speaking. The pharma industry's past is kind of littered with examples of bad outcomes, right?
Like what?
Severe side effects, drugs not working like they should. And often it boiled down to a lack of basic scientific knowledge, or maybe not fully appreciating metabolic effects, or just not pulling together the information that was already there. Think about things like enzyme induction, one drug messing with another's metabolism, or metabolic activation, creating toxic stuff, or, critical drug-at-drug interactions. These used to be huge roadblocks.
So the real challenge now isn't just gathering data, because we can get tons of it. It's more about truly understanding decades of science, mastering these complex tools, and then intelligently putting it all together to predict how a drug will actually work in a patient.
Precisely. That's the core task. A huge task.
Okay, speaking of those tools, the technology, you said it's like night and day. What's been the biggest single game changer? What really flipped the switch?
Easily the speed and just how common, powerful instruments have become. Things that took months, we can genuinely do them in minutes now.
Minutes? Seriously?
Yeah, like LCM SMS instruments, liquid chromatography, tandem mass spec. They're everywhere now, as common as, say, HPLC with UV detectors were maybe 15 years ago.
Wow.
And it's not just about speed for speed's sake. It's fundamentally changed things by embedding biotransformation deep into drug discovery much, much earlier in the process.
Okay, but all that power, all that data.
Yeah.
Surely that brings its own set of problems, right? Is there a risk of just drowning in data or maybe relying too much on the machine output?
That's a really sharp point. The dilemma isn't just having the fancy machine. It's the careful, thoughtful choice of which experiments, which technology you actually use for the specific question you're trying to answer.
Right. Tailoring the tool to the job.
Exactly. A good biotransformation scientist has to understand the limits of their tools. Specificity is great. It gives you sensitivity, but it can also blind you. It can filter out important stuff you didn't know to look for.
I see.
So often you need to use complementary techniques. Maybe come at the question from a totally different angle to clear up ambiguities or just, build confidence in your findings. Scientific credibility is key.
Okay, so technology supercharged things. But let's get to the core science. What are the foundational disciplines? Where does a biotransformation scientist really start?
You absolutely have to start with chemistry, no question. Biotransformation is a chemical process, fundamentally.
Makes sense.
So the most valuable studies lean heavily on analytical and bioorganic chemistry, NMR, nuclear magnetic resonance spectroscopy. Invaluable.
Why NMR specifically?
Because it gives you unambiguous structural assignments for metabolites. You know exactly what atoms are connected where. A really well-rounded biotransformation scientist needs to know how to use NMR, interpret the data, especially the more complex stuff like heteronuclear decoupling experiments. It tells you the precise structure.
So it's not just what changed, but really digging into how did this change happen chemically The mechanism.
Precisely. Exactly. You'll often hear the best biotransformation scientists described as good electron pushers.
Electron pushers. Okay. Unpack that for me.
It means they have such a deep grasp of bioorganic chemistry, they can visualize how electrons move during the reaction. They understand the mechanism of how molecules rearrange during metabolism.
Okay.
They can usually rationalize how a transformation happened mechanistically, and maybe more importantly, they can spot when a proposed metabolite structure just doesn't fit the chemical logic or the data.
Right. It just doesn't feel right chemically.
Yeah, exactly. And that shared language of bioorganic chemistry, that's often the bridge between biotransformation scientists and medicinal chemists.
Ah, the collaboration aspect.
Right. They work together to tweak molecules, maybe make them less likely to be metabolized in unwanted ways, reducing metabolic liability, or designing out features that might lead to those nasty reactive metabolites.
That sounds like a really powerful synergy, but also potentially tricky. Do they ever pull in different directions?
Oh, sure. Sometimes the medicinal chemist is laser focused on making the drug bind tighter, efficacy, while the biotransformation expert is worried about a metabolic weak spot.
The hot spot?
Exactly. Finding that balance potent and metabolically stable, that's the art. And this leads right into the biology side, because these transformations, they're run by enzymes.
Okay.
So to fully grasp xenobiotic disposition, how the body handles these foreign chemicals, you need to understand the specific enzymes doing the work.
So enzymology comes into play.
Big time. We can now study individual enzyme reactions, see how they fit into the bigger picture. You can ask what happens when enzymes are involved and how do they do it very, very quickly now.
And that speed, that understanding, must change how we use animal models to predict human outcomes.
It absolutely does. Hugely. We have ready access to cells, liver fractions, you name it, from humans and all the standard preclinical species.
Right.
So making those interspecies extrapolations predicting human pharmacokinetics from animal data is much more reliable. Unexpected PK performance in humans. It used to be a major reason drugs failed later on, drug attrition. Now, it's actually pretty rare.
That's a massive improvement in efficiency and safety.
It really is.
Okay, what about differences between people? We're not all the same metabolically.
Great question. Molecular biology has given us huge insights there. We can clone individual human metabolizing enzymes, express them, study them in isolation.
So you can pinpoint which enzyme does what.
Yes, and crucially, we now understand the genetic basis for many polymorphisms, those variations in enzyme genes that make people fast or slow metabolizers of certain drugs.
Ah, the pharmacogenetics aspect.
Exactly. So we can anticipate potential differences between individuals. We can actually predict if a drug might work differently or have more side effects in one person versus another.
So you could potentially screen patients.
We can. We can even design clinical trials to exclude people predicted to be poor responders, for example. And molecular biology also gives us relatively cheap ways to screen drugs early on to see if they might induce or inhibit key enzymes heading off those drug interactions before they become a problem.
Okay, let's tie all this fantastic science back to the ultimate goal. Making sure a drug is effective and safe. Even the simplest case, right? Say a drug just injected and passed out unchanged by the kidneys. Still things to consider.
Oh, absolutely, even then.
Right.
Is the excretion too fast? Is the drug cleared before it can really work?
Good point. Compromised efficacy.
Right. Or do you need to adjust the dose for someone whose kidneys aren't working well?
Renal impairment, yeah.
And could just having high concentrations of the drug hanging around in the urinary tract cause local problems, safety concerns there?
So the biotransformation scientist always has to ask, what could this mean? Not just what's happening.
That's the mindset. Always thinking about the clinical implications.
So flipping that around, in early discovery, how vital is getting that biotransformation picture early on for ensuring the drug actually works?
It's fundamental, honestly. A huge amount of effort and resources go into optimizing a molecule's disposition properties just to get the desired therapeutic effect.
Because once you've made the molecule,
its properties are pretty much set, yeah. You can't easily change how the body's going to handle it after the fact. So, getting the optimization done right during discovery is just critical.
trying to fix it later is inefficient.
Hugely inefficient. Just relying on trial and error pharmacokinetics isn't the way. You need that biotransformation input. Like a simple early study using HPLC with UV and mass spec detectors running in parallel, it can quickly show you the biotransformation hotspot.
The vulnerable parts of the molecule.
Exactly. And that tells the chemists exactly where they might need to make changes, make smarter modifications.
Okay, and the flip side, safety. Drug toxicity is Well, it's the big killer of drug candidates, isn't it?
It is, and think about it. Xenobiotic metabolism, by definition, is the body dealing with a foreign chemical. So a massive part of the biotransformation job is figuring out how metabolism turns off the good effects, the efficacy,
Right.
and how it sometimes leads to bad effects, the toxicity, things like how long a drug lasts, how intense its effect is, how much that varies between people, that's often tied directly to its disposition.
And that variation can be dangerous.
Especially for drugs with a narrow therapeutic index, that small window between effective and toxic doses. If metabolism varies a lot between people, some could easily tip into the toxic range, adverse effects.
And we touched on drug interactions earlier.
Yeah, a huge safety concern. Your drug might inhibit or induce enzymes that handle other drugs the patient is taking.
So if it's an inhibitor,
You could get dangerously high levels of the other drug, exaggerated effects.
and if it's an inducer,
It could chew up the other drug too fast, making it ineffective. A therapeutic lapse. Either way, it can seriously limit how a drug can be used.
Okay, you also mentioned reactive metabolites. That sounds particularly nasty. What's the key thing for, say, our PhD student listener to grasp about those?
Right, molecular activation forming these biologically reactive intermediates is probably one of the most intensely studied areas.
Why? Because they cause toxicity.
Often, yes, they can covalently bind to proteins or DNA. The good news is we have a pretty solid understanding now of what chemical structures, what structural features might make a molecule prone to forming them.
So you can predict the risk.
We can anticipate it, and once you identify that potential, medicinal chemists can often design around it, minimize or avoid forming that reactive species altogether through careful molecular redesign.
A crucial collaboration again.
Absolutely. Both biotransformation scientists and medicinal chemists really have to know this area well, because, as you said, toxicity is arguably the single biggest reason drugs fail. So the biotransformation scientist is almost guaranteed to be pulled into investigating toxicity issues. They need to apply rigorous science to figure out what's going on.
It's incredible how central this field is. Okay, so we've done the science, maybe we understand the metabolism, the safety profile, but the job's not done, right? How does all this work actually translate into getting a drug approved and protecting the company's investment?
Yeah, that's a critical bridge because, if a drug doesn't get registered, approved by regulatory agencies,
That helps no one.
Exactly. No therapeutic benefit, and the whole thing was just a phenomenally expensive scientific exercise. And getting a new chemical entity, an NCE, registered, it's getting tougher.
How so?
Regulatory bodies like the FDA, EMA, they're constantly learning, and they formalize those lessons learned into official guidances and regulations. The bar keeps getting higher.
So the data you generate in the lab has to meet these incredibly strict external standards.
Absolutely. It's not enough for it to be scientifically sound. It has to be collected and documented according to GLP good laboratory practice.
Rigorous documentation.
Meticulous. It needs proper precision, accuracy, and it all has to be woven together into a coherent, compelling story about the drug. It gets scrutinized from every angle to make sure there are no hidden flaws that could undermine the big decisions.
And all that work eventually gets distilled down for doctors.
Right. The ultimate summary is the product label. That's the document negotiated between the company and the regulators.
The package insert.
Exactly. And a really well-written label concisely explains the drug's disposition, especially any aspects that might cause big differences in how patients respond. For the biotransformation scientist who's worked on it for years, getting that label right is often their final masterpiece, their piece de resistance.
Leaving their mark on years of work. Okay, and the money side. Patents, intellectual property, it feels separate from the biology, but you're saying metabolism links in here too.
Oh, absolutely. Bringing a drug to market costs. Well, billions sometimes. It's staggering. Patents, especially the composition of matter patent protecting the molecule itself, are what give companies the confidence they might recoup those costs.
Makes sense.
But here's the twist. Disposition, especially biotransformation, can throw a real curveball into the intellectual property.
How? How can metabolism affect a patent?
Well, think about the sheer chemical creativity of metabolic enzymes. They can transform a drug into new structures, things the original inventors might never have even thought of.
Creating new molecules inside the body.
Exactly, and maybe one of those metabolites is actually the main active drug. Or maybe it's even better than the parent drug, more potent, fewer side effects, better PK.
Ah, okay.
Now, if the original development team didn't fully characterize these metabolites, didn't consider them in their patent strategy, someone else might spot the opportunity.
And patent the metabolite themselves.
Precisely, or develop it as their own drug. This isn't just a theoretical risk, it has happened. Many well-known drugs on the market today started life as metabolites of someone else's drug, marketed by a different company.
Wow. Okay, that's fascinating.
Yeah.
Can you give us some concrete examples? Real drugs.
Sure. These are classics, really important for you to know.
Okay.
Remember loratadine, widely known as Claritin.
Yep. Allergy med.
Its major active metabolite is desloratadine. That was later marketed as Clarinix.
Same mechanism, different drug product.
Essentially. Another big one, terfenadine, which was sold as Seldane. It got pulled from the market due to heart issues.
I remember that.
Well, its active metabolite was fexofenadine. That metabolite didn't have the cardiac side effects and became the blockbuster drug Allegra.
So the metabolite was safer and effective.
Exactly, a huge improvement. Think also about phenacetin, an older pain reliever. It's metabolized to acetaminophen.
Tylenol?
The very same. And one more. Hydroxyzine, an older antihistamine, is metabolized to cetirizine.
Which is Zyrtec.
Right. So these examples really drive home how critical understanding metabolism is, not just for science and safety, but for drug design strategy and protecting intellectual property. It's all interconnected.
Okay, that really clarifies the stakes. To help you, our listener, consolidate this, especially for those quails, here are a few questions based on our chat. Think about how you'd explain these concepts clearly, maybe like in a presentation or exam setting.
Good idea.
1. What were those 3 general categories of historical adverse outcomes we mentioned early on? The ones that really highlighted why understanding drug disposition is so vital. 2. We talked about the amazing new instruments. But beyond just having the tech, what's the main dilemma or challenge facing a biotransformation scientist using this technology today?
Okay.
3. Can you explain what we mean by a biotransformation scientist being a good electron pusher? Why is that skill so important?
The mechanism, right?
Exactly. And 4, can you name at least 2 pairs of parent drugs and their marketed active metabolites? The kind of examples we just discussed that show metabolism's impact on IP and drug development.
Got it. Good test questions.
Phew. Okay, what an incredible journey through drug metabolism. Seriously, from the nitty-gritty chemistry to the huge impact on safety, efficacy, getting drugs approved, even patent law. It's clearly a field that touches everything in pharma.
It really is. It's incredibly broad. And, getting the data, that's one challenge. But the real scientific step, the true mark of expertise, is turning that data into meaningful knowledge.
Making sense of it all.
Yeah. Asking the right questions beforehand, collecting the data the right way, integrating all the pieces correctly, and then applying that knowledge effectively.
So for you listening, as you dive deeper for your exams and think about your future research, what's the big takeaway here, the final thought?
Maybe it's this. How will you use your understanding of drug metabolism? Not just to describe what a drug does biochemically, but to really dig into why it behaves that way, and what are the wider implications for patients, for development. Who knows, maybe you'll be the one to spot the next big metabolite drug opportunity that others missed.
PharmaPal.